Hello friends, welcome to KNS Biochemistry. So in this session we are going to see about genomics and proteomics, one of the important area of genetics and bioinformatics. So we are going to see about genome maps and the different types of genome maps here. So in the background you can see a picture. It is the three important life critical molecules present in the living systems. One is DNA, the other one is RNA, third one is protein. All the three are important in central dogma. So what is this central dogma of molecular biology? It is having the combination of all the three molecules, DNA, RNA and protein. DNA can be duplicated into another copy. It is called DNA replication. So DNA can replicate. Then the information present in the DNA can be taken to RNA, and it is called transcription, where information coded in the sequence of base pairs in the DNA molecule is passed to molecules of RNA. Then the information which is there in RNA can be converted into protein molecule. This is called a translation, where information present in the RNA is passed on to proteins. It never passes from proteins to nucleic acids. That is, you see that protein to nucleic acids coming back is not possible. Only forward reaction from RNA to protein is possible. So all these three critical molecules, DNA, RNA and protein, are involved in central dogma of any form of living system. We are going to see about genome maps with this central dogma background. So what is this genome map or genetic map? It is a map that shows the relative location of two genetic traits, related or closely related traits. The way to do this is to use the offspring means F1, F2 generations of an organism, and track how many times two given genetic traits are inherited together. They are inherited from one generation to another generation. For example, hair color and eye color, both are inherited. There are two general types of genome mapping. One is called genetic mapping, the other one is physical mapping. Both types of genome mapping, actually these two mappings are going to help or guide the scientist towards the location of a particular gene or the section of a DNA called a gene on a chromosome. However, they rely on very different information. And if you go for positional cloning and gene mapping, we are having a lot of strategies. So we have general mapping strategies. One is called genetic mapping, other one is called physical mapping. Then we have RFLP, restriction fragment length polymorphism linkage analysis. In that we have RFLPs linked to disease genes, then predictive value of linked RFLPs. Then gene isolation with linked RFLPs, where we have chromosome walking, then criteria for identifying a gene. Then physical mapping. This is physical mapping of a particular gene, where we have cytogenetic analysis. Fluorescence in situ hybridization technique is there. FISH, fish is there. So we have general mapping strategies like genetic mapping. What is the definition of genetic mapping? By definition, the ordering of the genes on a particular chromosome or chromosomes according to recombination frequency. So here genetic mapping may sometimes be required first before probes can be found to use in physical mapping. Then physical mapping by definition says that determination of physical distances between the genes, adjacent genes in base pairs of DNA, that is the distance, using cytogenetic analysis and molecular techniques. Physical mapping is used to ultimately pinpoint or exactly locate the gene of interest. So if you take a human genome, Mapping the human genome can be done by all the three: genetic mapping, physical mapping, and DNA sequencing method. So we have classical genetic linkage studies. So genetic linkage mapping, it involves determining the statistical association of specific traits with genetic markers on chromosomes using pedigrees and crosses. So for that we have several methods used. So we use recombination frequencies to determine relative distances between marker genes on a chromosome. Then genome-wide association studies called GWAS is there. Then humans require 24 different maps, one for each of the 22 autosomes and one each for X and Y chromosomes. Then marker alleles are used to determine the rate of recombination. Example, crossing over is there between the linked genes. So linked means on the same chromosome or on the same genome. So the unit which is used or measured for each linkage map is recombination frequency. That is nothing but recombinants divided by total progeny. This is reported as map units, MU, or centimorgans, CM. Different types of markers, they are used in genetic mapping. So genes can be used as genetic markers, but they are not ideal choices because they occur frequently. Example, every 100 kilobase pairs in human genome we have a marker. So greater marker density, it is usually required for identifying the genes. So three major types of markers are used here. One is RFLP, restriction fragment polymorphisms. This is actually substitutions at a restriction site, where in the restriction site restriction endonucleases are going to cut the DNA molecule. Then STR, very short or short tandem repeats STR, otherwise called microsatellites. Then SNPs, single nucleotide polymorphisms. Then STS, that is sequence tagged sites. So genome-wide mapping can be done. Here you see that high density genetic mapping, it was first revolutionized in the year 1980s by the discovery of abundant polymorphic genetic markers like macrosatellites and microsatellites. Then research teams, they collaborated and they added to a common database. Then in the year 1994, human genetic map, it had localized almost 5,264 microsatellites to 2,335 chromosome loci. So now it's the average density of one marker for every 599 kilobase pairs. So in this process, thousands of sequence tagged site, that is STS, were identified. STS, that is nothing but couple hundred base pairs of known sequence. Two types of physical mapping, physical maps useful for sequencing a genome. One is called low resolution maps. They are cytogenetic or FIS sketch maps. Actually chromosomes can be stained with some dyes. So stained chromosomes, they produce banding patterns composed of bands that average 6 MB. Then regions are designated by their position relative to the centromere, which is there at the center of the chromosome. Q is equal to long arm, and the P, which indicates short arm, numbered from the centromere starting with the number 1. So genes and other sequences are localized to chromosome maps with probes, and by using a technique called fluorescence in situ hybridization, FIS. Various types of radioactive probes, radioactive isotope probes, and stains also can be used to mark specific regions of chromosomes. Then it provides a physical map of the overall structure of each chromosome or the region of the chromosome. Two types of physical maps very useful for sequencing a genome. The second one is called a high resolution map, YAC or BAC clone contig maps. So mechanically shear or partially digest genomic DNA with restriction enzymes and clone large 200 to 500 kilobase where see that kilobases overlapping fragments to Yorks or BACs. So an entire genome or single chromosome can be represented in a York or by BAC clone library. It depends on the starting point where the gene starts. Then overlapping YAC or BAC clones can be assembled into a scaffold without sequencing by DNA fingerprinting using markers like microsatellites. So BAC vectors with a capacity of 300 kb and ability to replicate in a bacterium called E. coli, Escherichia coli, that have become popular for genome sequencing. Now we see that routinely sequenced using the shotgun method. Sequencing the human genome, here we have two major players. One is called SGP, other one is called CRA. What is SGP? Human Genome Project, publicly funded international consortium like NIH, DOE, etc. Then Francis Collins National Human Genome Research Institute, NHGRI. Then began in US in 1990. They started with a goal of 15 years. Then genetic and physical mapping approach and dideoxy sequencing was done. Then Celera Genomics Corporation, CRA, they did it. They had a spin-off of Applied Biosystems API. Then J. Craig went our CEO. Then they created this in the year 1998 with a goal of 3 years. Direct shotgun method and dideoxy sequencing along with HGP's maps for validation is done. Both groups collected blood and serum and sperm samples from anonymous male and female donors of different ethnic backgrounds. So for identifying genes in the DNA sequences, we have to do first step, that is called annotation step. That is nothing but identification and description of putative genes and other important sequences. The second one is called ORFs, open reading frames. So this ORF is nothing but potential protein coding sequence that begins with a start codon and ends with a stop codon. So all ORFs come in all sizes. So not all ORFs they encode proteins. 6 to 7% they will not code for protein that is done in yeast. Then ORFs with introns, that is called intervening sequences, can require sophisticated computer algorithms to detect it. Then sequencing the human genome. Two major players. We have the HGP and CRE, as I mentioned earlier. And identifying the genes, we are having annotation step first, then ORF second. Then in HGP we have got milestones. In the picture you can see the former president of USA, Bill Clinton. So the year 2000 was the milestone where the work was completed on HGP. So 26th June 2000 where you see that in the White House press conference, the former president of USA, Bill Clinton, met the press regarding this HGP. So HGP started in the year 1990. Approximately 32.1 billion nucleotides of sequence data were sequenced. 7-fold coverage was done. Unfinished, that is 24% were completely finished. 50% near to finished. Now it is finished. Then Celera, they started in the year 1998. Approximately 14.5 billion nucleotides of sequence data were sequenced. 4.6-fold coverage was done. Complete assembled genome with 99% coverage was done. So the first assembled draft of a human genome that was simultaneously published in a scientific journal, Nature and Science, on 15th and 16th February 2001.